Hi. In today's lecture, we're going to talk about experimental design, one of those really key parts of being a scientist. Scientists design experiments all the time, and so it's necessary to make sure that you can design quality experiments, and so you get the results that are actually meaningful and you can interpret correctly. So to begin with, what I would like to do is talk about some of the vocabulary you've probably heard previously, but kind of a reminder. The first thing, the scientific method. The scientific method is a process scientists use in order to answer questions. Remember, one of the challenges scientists have is they're often asking questions no one knows the answer to. So the scientific method provides a framework that we're able to go through to get and work toward answers and hopefully identify possible errors along the way. The basic steps of the scientific method include starting off by asking a question. I wonder why something happens. So we ask a question, and then the next step is to do some research. It's very possible someone has already figured out the answer to your question, in which case, why bother spending time conducting experiments? So you do some research, internet, scientific journals of what have people already discovered. Then, if your question is still valid and you still want to pursue it, you're going to develop a hypothesis. Hypothesis is kind of an educated guess about what's going on. Next, you're going to design an experiment to test that hypothesis. The sole purpose of the experiment is to test the hypothesis. Is the hypothesis correct or incorrect? You then conduct the experiment, collect and analyze the data, and then draw conclusions about your hypothesis. Was your hypothesis correct or was it incorrect? And then one step that is often left out of the scientific method, but is very, very important for professional scientists, is share your results. Can you imagine if Galileo, Darwin, Mendel, any of these famous scientists who have completely changed their fields didn't share the results when they figured something out? Science progresses because scientists share their results and future scientists are able to build on that knowledge to create something bigger and better. So it's a constantly growing process. The final part of the scientific method is to repeat. So when you do the scientific method, often your results say you were wrong. So your hypothesis is not valid. Okay, you have to reject it. So something else must be going on. And so you go back and you reevaluate and you go through this process again and again, trying to chip away at the unknowns until we can actually understand what is going on. So that's the basic scientific method, and you use this every single day of your life. Let me give you an example. If I have a flashlight that's not working, my question might be, why is it not working? So I might do a little bit of research into flashlights, think about what I already know, look up online, find YouTube, and I come up with some ideas. And what are some possible reasons that my flashlight might not be working? Well, maybe the batteries are dead. Maybe the light bulb burnt out. Maybe some of the electrical wires aren't connected properly. All of these are possibilities, but I need to pick one to focus ahead. So I'm going to go with a hypothesis about the batteries. And my hypothesis needs to be testable. So I'm going to say, if the problem is the batteries, then changing the batteries should fix it. Now I'm going to design an experiment. I'm going to get new batteries, and I'm going to conduct it by putting them in the flashlight and turning it on. If the flashlight turns on, fantastic. My hypothesis was correct. It was the batteries. If my flashlight does not turn on, it doesn't mean I wasted my time. I've eliminated an option, and now I'll go through this process again in order to figure out what is the problem. That's the basic scientific method. So we use that every single day, though we often don't consciously break it up into those sections. But as scientists, we need to. So scientific method is very important. Another term or series of terms that you're going to hear when we talk about this is opinion, hypothesis, and theory. An opinion is an individual's thoughts about some topic. It does not need to be based on anything. It can be based on facts. It can be based on totally made up information, something they read online. There's nothing testing the validity of an opinion. You have opinions about topics. I have opinions about topics. They might be the same. They might be different. Scientists don't work in opinions. We have opinions, but that's not science. Science is about backing up your ideas with evidence. And so scientists frequently use hypotheses. A hypothesis is an educated guess that's based on previous knowledge and is testable. So where an opinion, that sucks. That's more of an opinion. How do I test that? I can't. A hypothesis, that product is not good because of this. Well, I can go and test those things. So a hypothesis must be testable and is generally based on previous knowledge. As scientists, we're interested in hypotheses, not opinions. However, you also have to be aware when you hear someone's opinion that some opinions are more valid than others. For example, we are talking about a global pandemic, and I'm talking to a neighbor, and they have opinions about the pandemic, and then I am talking to Dr. Fauci, who's been on the news multiple times. He is a certified expert in pandemics and stuff. His opinion carries a lot more weight with me than someone else who doesn't have his background. But it is still an opinion until he backs it up with evidence and studies. Finally, we come down to theory. A theory is an explanation about how something works that is supported by a large amount of evidence. It takes time to develop a theory. A hypothesis does not become a theory at the end of an experiment. A theory is something that is supported over and over by thousands of experiments over extended periods of time. For example, the theory of gravity. Okay, the theory of gravity. There have been so many countless experiments over hundreds of years testing gravity, how's it work and everything, that it has become a theory. We don't doubt it. Theory is about as close as scientists will ever get to saying truth or fact. Scientists don't deal in truth and fact. We deal in evidence. And a theory is about as close as we get to saying, so we have proven something. One other term you won't really hear in biology, but you do hear in other sciences, a law. A law is kind of like a theory, but it's much more narrow in scope. A law explains what will happen when you do something, but not why it will happen. So, for example, the law of gravity says, when I take this pin and let go, it will fall. The theory of gravity explains why it's falling. So theories do not become laws. Theories are the highest level of evidence that we have in science. And so when you hear a scientist talk about a theory, they are very, very confident in what they're talking about, as opposed to the general public tends to use the theory the way scientists use hypothesis or opinion. Theory is backed up by a tremendous amount of evidence. You'll hear those terms thrown around quite a bit over this course, so I want to make sure we clearly understand each of them. When you do science, you're often manipulating and looking at different variables or different measurements, things you're looking at. And the two main types of variables we deal with are independent variables and dependent variables. So let's start with independent variables. So independent variables, these are variables that are not impacted by the dependent variable. In other words, the dependent variable has no effect on the independent variable. Okay? An example of this, if I am looking at rain affecting plant growth, it makes sense that the amount of rain affects the amount plants grow. But at the same time, in reverse, the amount plants grow does not affect the amount of rain. So the amount of rain would be our independent variable. Okay? The dependent variable, on the other hand, is the variable that is impacted by the independent variable. And there's a very easy way when you look at an experiment to know which one's which. If you are looking at an experiment, the independent variable is the one that is being manipulated. So our plant rain experiment, we're changing the amount of rainfall plants get. So we're manipulating the amount of water. That would be the independent variable. The dependent variable is the one that you are measuring as part of the results during the experiment. That's the data you're collecting to draw conclusions from. So independent variable, you're manipulating and changing it in the experiment. Dependent variable, you're recording it as your results. The AP test periodically has questions asking you to identify dependent and independent variables. So it's important you understand these terms that we're able to apply them throughout the course in different experiments. So as you design experiments or conduct them, ask yourself which variables are independent and which are dependent. Another thing you've likely heard about is a control. So what exactly is a control? So here's an experiment being shown where we are putting some biofertilizer on plants. So imagine I go out into a field and I add some biofertilizer onto the plant. Let's say it's in my garden. So I put biofertilizer and the plants grow. Am I confident now saying that the plants grew because of the fertilizer? No. It is possible they grew because of the fertilizer, but maybe there were a whole bunch of other things that were there that caused them to grow instead. I can't say with confidence that the sole reason they grew was the fertilizer. So in that case, I need to create what's called a control group. A control group is a group that's treated the exact same way as the experimental group, except for one variable, our independent variable. So in this case, I would have two groups of plants. One of them I gave the biofertilizer to, the other group I gave no biofertilizer to, but I treated them the exact same other than that one variable. That way, if the experimental group grows more than the control group, I can say with some confidence now that it was because of the independent variable, not some other factor. So generally, it's very important to have a control group as part of your experiment to say with confidence that the results are a consequence of that particular independent variable. And finally, statistics. You might have heard of statistics, you might have taken a class in statistics, or you might be taking a class in statistics this year. The good news is I will teach you all the statistics you need to know. You don't need to know a lot of it, but statistics is simply a way to mathematically organize data to make it easier to understand trends and patterns. So statistics are commonly used by scientists to analyze their data, and we'll get into that in a future lecture. Okay. So designing a research study, I've got some vocab terms out of the way. So here's a couple points when you're designing a research study you'd want to take into account. And you'll see these are pretty closely related to the points of the scientific method. First off, you want to identify variables of interest and the population of study. So what dependent and independent variable are you looking at? And also, who are you studying? You need to figure out what group is your question about. Are you asking a question about students at Culver? Are you more narrow than that? Students in biology classes? Or broader than that, students in Indiana? So you need to decide what population you're looking at because that's going to influence what data you need to collect. You need to come up with a detailed plan for collecting your data. You don't just go out and try to grab numbers any way you can. You need to make sure that you're collecting data in a systematic way that you can repeat over and over. You also need to make sure that the data you're collecting actually represents your population. Is your sample truly part of your population? If I wanted to know the favorite dining hall food of CMA students, would it make any sense for me to go out and survey CGA students? No, it wouldn't, because that would not answer the question I'm trying to identify. You have to collect the data. Make sure to record all your data. You need to describe your data. You'll end up with a bunch of numbers. How do you actually analyze? And we frequently use what we call descriptive statistics techniques. For example, averaging, standard deviation, standard error. We'll talk about those later. You need to interpret the data. And often you'll need to use another branch of statistics called inferential statistics to try deciding, are the changes or differences I see between these two groups because they're actually different, or is it just random noise and slight variations that are making them look different? And statistics can help us answer that question. Again, more on that later. And then finally, as a scientist, you want to identify possible sources of error, things that might have made your results, make you less confident about your results than you would be otherwise. Does your sample truly represent that larger population? These are questions scientists have to ask because they're trying to draw conclusions. And so identifying errors is an important part of what we do. When we collect data, there are several different ways to collect data. And 4 different primary study types that we're going to talk about for a minute. The first one is an observational study. So observational studies, this is when a researcher observes and measures data, but does not actually change the environment at all. So they're just kind of an outsider looking in. And an example of this would be a researcher who's looking and recording what children who are 3 years old are doing with non-food objects. When they pick something up, do they put it in their mouth or not? Okay, that might be an observational study. The researcher's not asking the children anything, just sitting back and watching what they do. The second type of study is an experimental study. An experimental study is where a treatment is applied to part of the population and responses are observed. Okay, in these studies, you often have a control group, and sometimes you have what's called a placebo group. We'll talk about that in a minute. Okay, so an example here is someone who's diabetic is going to take a pill to see if it helps reduce their risk of heart disease, while a control group is going to take a water pill that doesn't actually do anything. And does the pill have any effect? Experimental studies are the main kind of studies we tend to do in AP Biology. Back in our lab, we're going to actually manipulate things and such, so you'll see those pretty frequently. Two other types of studies we can do. A simulation. A simulation is a mathematical or physical model that's used to reproduce conditions of a situation. These experiments are done when the experiment would be too dangerous to do. For example, when they test car safety, they use crash test dummies because they drive cars at 60 miles an hour into a wall. No person is going to volunteer to be part of that study because death is a very high likely possibility. And so they simulate people with dummies, okay? Or another option would be if an experiment is too costly or it's just not feasible to do. For example, a lot of the work with global warming, we only have one planet to do. So it's hard to, let's do global warming on one planet and not on another planet. Well, we don't have another planet. Or to do something on that large a scale might cost way too much money. And so they will create computer simulations and such to try to mimic what would happen if a certain effect was done. Now, simulations can be very, very effective, but the simulation is limited by how much information and data and how accurate your simulation is. So that's always a question. Scientists are constantly trying to improve the accuracy of their models and their simulations to draw better and better results. Surveys. Actually, going back to simulations for a second. This year, COVID-19 pandemic, if you've been watching the news, you'll hear estimates of this many number of people are going to catch it, this many number of people are going to die, and then a month later you'll hear completely different numbers, and a month after that, different numbers again, sometimes going up, sometimes going down. What is going on? Are they just guessing? No, they're modifying the simulation as we learn more. In the early last year when COVID just started showing up, we didn't know much about it, so we had to make a lot of guesses. Since then, we've learned more. We find out how contagious it is, how many people tend to get passed on. Masks are introduced. Okay, what effect does that have on our simulations? And so on. And so the predictions are modified as the simulations have become more and more precise. Our fourth way to collect data is using surveys. Now, in biology, sometimes we do this, but psychology and other branches of science use surveys pretty frequently. So this is an investigation of one or more characteristics of a population, usually conducted by interview, through mail, over the phone, or online. For example, a survey conducted on female physicians to determine whether the primary reason for their career choice is financial stability. You can think of many different types of surveys. In fact, you might have participated in surveys. They're collecting data to learn more about their population. Okay, so let's see if you understand and can apply these four different types. So here, I've got four different studies. What type are each of them? Are they a simulation, a survey, an observation, or an experiment? Okay, so I recommend pause the video for just a second and make a guess of what kind each of these is. I'll wait for just a moment. Okay, let's see if you're right. So our first one, a study of the effect of changing flight patterns on the number of airplane accidents. I really, really hope you put that would be a simulation. We do not want to go in there and actually start changing flight patterns and see if planes crash into each other. We want to do this on a computer and make sure no one really dies. So that would be a simulation is the best way to study that. Next up, a study of the effect of eating oatmeal on lowering blood pressure. That would be an experiment. We would probably have a group that eats oatmeal and one that doesn't eat oatmeal, and we'd see what happens to the blood pressure. A study of how fourth grade students solve a puzzle. Okay, this is going to be an observation. Just sit back and watch how they solve the puzzle. Don't give them any clues, just see what they do. And finally, a study of U.S. residents' approval ratings of a U.S. president. These are done all the time, and this would be a survey. Hopefully you were able to get all of those correct. Okay, so now let's take a look at some of the things that scientists really need to take into account when they're designing not just an experiment, but a well-designed experiment. One of the biggest problems we have when we try to identify factors is when the researchers can't really tell which variable had an effect. Okay, we see a change, but what variable had it? For example, a coffee shop owner wants to attract more customers to her shop, so she decorates it in bright colors. At the same time, a brand new shopping mall opens across the street. If business at the coffee shop increases, can you determine if it was caused by the new colors or if it was caused by the shopping mall? You can't. You don't know. It could be caused by either one. So if she drew a conclusion that, oh, it's because I painted bright colors, that's why business is up, she might be completely wrong. What's going on here is we have too many variables. Scientists are very careful about this because when they design an experiment, they want to be testing only one variable. And that's why you have your control group. Your control group and your experimental group are treated the exact same, except for the one variable, or independent variable that you're going to change. Everything else should be the same. So any changes you observe, you could say with certainty was because of the independent variable. So making sure there aren't multiple variables changing at once is important when designing an experiment. Another challenge that scientists, especially scientists who study humans, have realized is problems can occur when the subject knows they're part of an experiment. And this is called a placebo effect. In medical studies, they have shown pretty conclusively that the mind is a very powerful tool when trying to get better. For example, if they take a group of people who are ill and they give them a sugar pill, so it's a pill made out of sugar that has no medicine in it at all, and say, hey, this is medicine that's going to make you better, and they give it to them, a surprisingly large number of people will feel better, even though they didn't take any medicine, because they believe they took medicine, it has an effect. We call this the placebo effect. And so scientists will use a placebo, a false treatment that looks like the real treatment when doing, for example, medical studies. Like if they wanted to test if giving medicine helps depression, they would give the experimental group the medicine, and they would give the control group, our placebo group, a sugar pill. And if both groups get better, that shows the medicine's not making them better. Instead, the belief they're going to get better is helping them get better. On the other hand, if our experimental group that took the medicine shows greater improvement than our placebo group, then that suggests that the medicine actually is having an effect. Now, here's the thing. In AP Bio, we actually don't deal with placebo very often because our experiments we're doing are on people. And animals and other things like that don't tend to know they're part of an experiment, so we're not worried about a placebo effect. But I wanted to introduce the concept in case you ever hear it. Randomization. When you're designing experiments and you get your subjects that you're going to experiment on, you need to randomly assign those subjects to different groups. In an ideal world, they're completely random. Okay, if I were to go and grab the students at Culver Academies, I would go ahead, let's say I grab 800 students, almost the full student body. And I'm going to break them into two groups, a control and experimental. I should randomly assign each of them. Half of them go control, half experimental. Okay, so randomly decide that. Or maybe I need to subdivide the groups into blocks. So even though I'm looking at Culver Academies, I'm going to break them into freshmen, sophomore, juniors, and seniors. But within each of those groups, I'm still going to randomly assign experimental and control groups. So you need to be careful about not randomly assigned because biases can show up that are going to impact your data. For example, if I say, okay, I'm going to go ahead and assign these two groups, 10 people. The first 5 people to walk through my door are going to be in group A. The next 5 people are going to be in group B. And the first 5 people walk in, they're in group A and group B. The problem is the 5 people in group A are all members of the same family. And so maybe they share more similarities than the average person does. And so that might bias your results somewhat. Another example given here is an experiment testing a weight loss drink. You can create blocks for subjects who are 20 to 29 years old, 30 to 39, and 40 to 49 years old. Then in those blocks, randomly pick people to be in the treatment group and the control group. Okay, sample size. The goal of any experiment is to draw inferences about a larger population. Okay, and how confident are you your sample, the population you looked at, actually represents the larger sample? So, for example, let's go back to students eating at the dining hall. Okay, I've got a student who wants to eat at the dining hall. I say, what's your favorite food? And they tell me beef tacos. How confident are you that beef tacos represents the favorite food of all students at Culver? Okay, um, probably not very confident. On the other hand, if I go and I survey 400 Culver students and 80% of them say beef tacos, now how confident are you that beef tacos truly represent the most popular dish at Culver? Probably a lot more confident. You can think of sample size kind of like Amazon product reviews or Rotten Tomato movie reviews. If you go into Amazon, you're looking at buying these new headphones, and one person has reviewed them and gave 5 stars. Best headphones I've ever had. How confident are you that those are the best headphones ever? Probably not really. I mean, maybe that's the person who invented them or someone trying to sell them. On the other hand, if there are thousands of reviews and the vast majority of them are 5 stars, you're much more confident this is a good product. Okay, so large sample size are ideal. However, you also need to be aware that there's a limit in time and money to how large a sample size you can get, the logistics. So there comes a point saying large enough as far as your sample size goes. And one of the challenges we deal with a high school biology class is often our sample sizes are fairly small, but they still give us data to play with. Okay, experiments should be repeatable, and you should expect if you do an experiment over and over, you should see similar trends and patterns. If you don't, something's going on with your results. And so one of the ways to increase the chances of getting the same results is having larger sample size when you do the experiment. Okay, let's take an example. I want you to try determining what's wrong with this experimental design. So a company identifies 10 adults who are heavy smokers. 5 of the subjects are given a new anti-smoking gum, and the other 5 are given a placebo, another gum that's not anti-smoking. After 2 months, the subjects are evaluated, and it's found that the 5 subjects using the new gum have quit smoking. Fantastic. Our gum, it works great. Awesome. We should start marketing this, spend billions of dollars, and sell it all over the world because it cures smoking. Right? What's wrong with that conclusion? Okay, if you need to think about it for a sec, go ahead and pause the video and then come back and we'll talk about it. There are 2 main problems with this. First off, the first problem is sample size. Our sample size is really small. So I've got a very small sample size. Okay, so a really, really small sample size. Am I confident that this is going to be true for all heavy smokers? Not really. I need a larger sample size to verify that. So I would want to repeat this experiment with a larger group. The second problem, what are the results for the placebo group? We don't know. It doesn't give them to us. Maybe all 5 members of the placebo group also stopped smoking, in which case it wasn't the anti-smoking gum. Okay? Without giving us all the results, I'm not confident in any conclusions that might be drawn. One more example. A company identifies 1,000 adults who are heavy smokers. The subjects are divided into blocks according to gender, male and female. Females are given the new gum and males are given the placebo. After 2 months, female group has significant number of subjects who have quit smoking. Again, pause if you need to. So what's wrong with this particular experiment? Our first key problem, something new, is the groups are different. It's true that we have an experimental and placebo group here, control group, but one group, the experimental group is all women, and the placebo group, the control group, is all men. Maybe the ability to stop smoking is gender dependent. So I've added an extra variable here besides the gum. So I'm not as confident in any conclusions I might draw. The second thing is we don't know the result for the men. We don't know the result. So again, we cannot draw any conclusions. Maybe they also showed significantly fewer smoking now. Okay. In AP Bio, you will be designing experiments and you will be drawing conclusions about experiments. You will also be analyzing experiments to see if they are well designed. Many experiments are not, especially ones that you hear referred to in the public. So my hope is this is an introduction. We will continue to develop these skills throughout the course as we do experiments, but you need to remember the various vocab terms and these concepts so that your experiments aren't poorly designed. Okay, hope you learned something, and I look forward to hearing from you on the next lecture. Take care.